Joining us for our workshop today on Good Laboratory Practices Light. We've partnered with Reproducibility for Everyone for this workshop. We're grateful for Harinder for introducing us to R4E, and I think I will hand it over to them to get started.
Great. So hi, my name is Nafisa and I'm joined with April. Who will be your instructors for this workshop. So welcome.
Welcome.
Just a few points. April is showing the slides that we have, and you guys have access to the deck. We also have some shared notes for the workshop, so feel free to go to that link and you can add comments and questions as we're working through the workshop. And then there's a handout, so feel free to go to those links as we go through the workshop. And then if you have questions, feel free to type them in the chat. I'm going to open that up so I can see it. But yeah, we'll get started. So, okay, I'll, I guess, introduce myself a bit more in detail. My name is Nafisa, and I'm an assistant professor at Midwestern University and at the University of Arizona. So I'm located in Phoenix. So I'm going to tell you a little bit about what R4E is. So next slide, please. So we are a group of scientists that are both in academia as well as industry. And what's brought us together is that we want to improve reproducibility of science. So we are an international group of people. We have people in the US, in Australia, in Asia, in Africa, that all over the world that want to really improve science in all fields. And so what we do is we work on developing the workshop that we're giving you today, so the content for that, as well as applying for funding, hosting these workshops at conferences, at universities. We've also, this workshop is open access, so it's all available on our website, and that website link is just on this slide. So we're really working, our main goal is to improve how science is done in terms of reproducibility. Okay, next slide. We do have several sponsors that make this possible, and they're listed here. And you know, there's been a lot of people that have been involved in our initiative. And Benjamin, who's pictured here, was one of the founding members of R4E. He's currently a professor in Australia. Okay, next slide. We've also documented or written about what we've done in terms of our initiative and all of sort of the aim. And that publication is available on Open Science Forum. It was also published in eLife as well. There are some initiatives that we're a part of in terms of reproducibility. A couple of our members are part of a reproducibility in neuroscience special topics in Frontiers of Integrated Neuroscience. So we're accepting manuscripts. I believe our deadline has been extended into early next year, so 2023. And so if you're interested in that, there's a little blurb about that. So just to go over some of our guidelines in terms of participation in our workshop, this is just a summary of kind of our expectations. And you know, if you feel that something has gone, or if you have any concerns, these are ways that you can bring that to either April or I's attention, or to myself's attention. Okay, great. All right. I sort of went over this, but we do have our notes document. So you can feel free to communicate with us through that. And so, yeah, and April's showing that. So this is live. You can add as we were going through the workshop. When we split it down, April's broken it down to the different parts of the workshop that we're going to go through. So feel free. And we also have questions for you guys in this notes document in terms of just some polls. All right. Any questions or comments thus far? You can just type in the chat or in the notes document. Let's give a minute. Okay, great. All right, so then we'll get on to the nitty-gritty of the workshop. So why does reproducibility matter? And why does it matter to you? And we did a pre-survey workshop. Prior to this workshop, we did a pre-survey. And one of the questions that we asked was, have you ever had problems reproducing your own work or someone else's research? And this is summarized here. So we see that your guys' response in terms of the percentages here. So someone else's work, there's been some issues, as well as with people's own research, or both, well, actually, I'm not sure that both is applicable here. Or April, do you mind chiming?
So when I summarized this, I had a hard time making this a bar chart that actually reflected the survey. So my apologies, but the both means that the person has had a hard time reproducing their own research and someone else's research.
Thank you. So it appears that, there have been some people attending this workshop that have had problems reproducing others or their own research. So next slide, the goals of this workshop are to describe the reproducibility framework. So in terms of the bigger picture, and then also provide you guys with some ideas, so some tools to make your research more reproducible. We'll go over a number of different types of tools that are available, and starting this kind of lifelong journey in making your research more reproducible. I was part of this workshop, I think maybe 3 or 4 years ago, and I learned a lot. Like you guys hopefully will too. And it can be a little bit overwhelming, but taking at least one thing or one tool and trying to implement that into your research can be a good starting point. And then as time goes, you can look at other tools and other techniques in terms of making your research more reproducible. So next slide, what we'll be doing is we'll be talking about what does reproducibility mean? What are the different modes of reproducibility? And is it only, doing good science, does that mean that it's only just reproducibility, or are there other factors? And then we'll talk about the different tools. So in terms of organization of data, documentation, analysis, and then dissemination, so sharing your results. Okay. So next slide. So what does reproducibility mean? So next slide. What reproducible research is a way to provide all the different things and materials, code that were used in a certain experiment, and being able to run that analysis again, recreating the results. A replication or replicating results means, taking, collecting new data and doing the same sort of protocol, and then doing that data analysis and then coming to the same scientific findings. So that's how replication and reproducibility differ. Next slide. This is a nice table that goes over the different modes of reproducibility. So we can see on the left side, you have the methods. So in terms of the same methods and then the same data, you get that reproducibility. When you have different data but the same methods, you get that replication. When you have the same data but different methods, you have this robustness of your results. And then when you have different data and different methods, you get this ability to generalize and make generalizable statements. Okay, so is it reproducibility all that matters in terms of doing good science, like rigorous scientific research? No, I think we can all answer that. There's a lot involved in doing rigorous scientific research. You know, there's the whole idea of doing the right statistics, looking at experimental redundancy, and all those different components that are listed in here. And so reproducibility is just one component of this, of rigorous scientific research. And so we can see in the next slide that there are different components to doing good, rigorous scientific research. And, you know, no one's perfect. We're all learning. I think being a scientist, you're always learning, even if you're a faculty member or a PI that has been doing research, you know, for a long number of years, you're still always learning. Okay, so what factors are involved in reproducible science? So there are a number of different factors. So next slide, in terms of how they impact reproducibility. So we can see that there are some factors listed here that decrease reproducibility. So there's things like technical factors, human factors, study design and statistics, as well as reward and incentives. And you can see that they all overlap and they're all interconnected. And if we go to the next slide, in terms of the technical factors, we can see that there are a number of different components to this area. You know, bad reagents, false cell lines, batch effects, depending on your area of research. And then if we look at the next slide, in terms of human factors, there's a number of different subcomponents that play an important role in decreasing reproducibility. So record keeping, data sharing or sharing of materials, reagents, methods, code. And the next slide looks at study design and statistics. And you can see, you know, we have things like p-hacking, harking, selective reporting, and you can see that these overlap with the other factors as well. And then in terms of the rewards and incentives, or sorry, then there's these mistakes and lack of training that are made sometimes that do play an important role in decreasing reproducibility. And then in terms of rewards and incentives, we also see that there are, you know, some of the incentives in scientific research, you know, the hyper competitiveness, the pressure to publish, to get novel data that's significantly different, you know, and then, you know, this whole going back to selective reporting, right? We only report data that's different, or, you know, we don't report a lot of negative data. There's also these paywalls. There's been a lot of awareness or increase in fraud, scientific frauds. If you guys are aware of like retractions for papers, there's actually a blog that I subscribe to, retractions.com. I believe that's what it's called, and they'll summarize the number of different retractions for that week in terms of journals and scientists. And so there's a lot of awareness now about fraud in scientific research. So I think this is a good slide in terms of summarizing all the factors that play a role in decreasing reproducibility. And so our goal with this workshop is to try and give you guys some tools that may help you avoid some of these factors. So next slide. So, you know, we've made a lot of progress in terms of scientific findings, and we do have, you know, different challenges that have come up in terms of reproducibility for different fields. But I think in terms of what, you know, has been said, and it can be summarized here, you know, we've done a lot and we can expect a lot in the future in terms of our advancement of scientific knowledge. Okay, great. Next slide. Okay, great. And so the goal of this project or this workshop is to show you guys some tools that can help accelerate science and hopefully propel you to do more reproducible research. Okay, great. So next slide. So we're going to talk a little bit about the potential for growth. And so and the next slide. So I believe this is some results from the survey that you guys took in terms of improvements that could be made to lead to more reproducible science. We can see the summarized here, the results summarized here. So better and more detailed methods for sure. Open data and metadata, increased statistical power, less pressure to publish. Yeah, that would be great. And to spend more time thinking about the results and fewer incentives to be first, better reagent sharing. So we're going to talk a bit about that, as well as better sharing of code. So next slide. So we're going to go over some tools and some resources, and you know, there's going to be a lot of content that April and I will cover, but think of this as, you know, sort of your starting point. If you are starting to think about reproducibility, some of you might have already, you know, implemented some of these tools, but you know, everyone starts somewhere and every little bit helps. In 2019, I started my own research group, and I made a point to incorporate some of the things I learned from this workshop, and that was the incorporation of electronic lab notebooks. So my lab has an electronic lab notebook, which I think helps a lot in terms of sharing protocols, sharing what people are doing. So, you know, every little bit helps, and no one is perfect. We're all learning. Okay, great. So next slide. So, you know, we're sharing a bunch of tools with you guys today. So, you know, thinking about how some of these tools incorporate into your research, some of them might not be applicable at all, and some might be really applicable. So, you know, keep that in mind as we go along. And I'll pass it over to April now.
Awesome, great. Thank you, Nafisa. So my name's April, and I'm going to go into the data management section, which is a really, I think, oftentimes an underappreciated aspect of reproducibility. And sort of following up on what Nafisa just said, when we're focusing on those things that will help us in our research make our lives less painful and more organized, that is a great place to start for reproducibility, because when we are organized and can understand and remember what we did and what our lab members are doing, all of that feeds into having more reproducible work when we go to disseminate that in the future. So data management is a great place to start. So we're all familiar with these feelings associated with managing data sets. We can't find the file. We don't remember where the most recent version is. We don't remember which version is the most recent version or what changes were made. We might not have a system for naming files, which makes it hard for you to identify without opening it and exploring it. One of the things that happens a lot of times is you don't remember, well, is this my raw data or have I touched this? This panic, these questions familiar to all of us, and we're going to help you feel less of that and feel more confident about how you are managing your data. So the most important message is to plan ahead. It really pays off to do some of the legwork up front. What you want to do is think about the types of data that you're going to get in a particular project, to think through how best to structure your workspaces that you're going to be using in your research to keep that data organized. So what that usually means is to create a simple data management plan. These are sometimes called DMPs. Some of you might be new to the idea of a DMP. Some of you may have written DMPs before. Everyone will be writing DMPs in the future. There's going to be more and more requirements from funders such as NIH and NSF to include a data management plan. And what I would encourage you to do is to not think of that as a task or a chore that you must do in order to receive that grant, but to reflect on how this is actually going to help you think through how to manage your data so that it is less a source of stress and more a way of saving time and working more efficiently on your project. So you're not blindsided at the end by a requirement to have documentation or a plan that you didn't think through from the start. So there's this thing here called the DMP tool, and this is a great place to start when you're wondering what a DMP is. It is a way of building a data management plan, a little bit like a template that can help you think through the types of information you want to put into that plan. And one of the benefits to it is that they have a lot of examples of data management plans associated with different funders. So that can give you a head start in getting a good DMP put together. So feel free to share in the shared notes document or in the chat experiences that you've had trying to plan your data management strategies. If you've used the DMP tool before or another tool to help you plan your data management, feel free to share that. This is a workshop where we encourage people to share what's worked for them. And don't try and do everything at once. Start simple. Think about the high level questions that you need to explore before you dive deeply into what specific tools you might use. So let's go to the next slide and see what some of those higher level questions might be when you're approaching data management for a new project. You want to think about what you're producing as part of the project, and then think about the different types of data and how they will be organized, how they'll be documented, what standards exist, and will you be adopting those standards? Where will things be stored? How will your access to the data be protected and controlled? And also the flip side of that, how and what will be shared, and then beyond the life of the research project itself, how will it be archived and preserved? So for each of these questions about the life of the data sets you're going to be building, you have to think about who should be taking responsibility for those activities. To think that out in advance, because that might dictate different resources or people you need to contact. And then the timeline for when these activities need to happen. And that helps you think ahead about if you are going to be sharing it, when are you going to be sharing it? That's an important thing to think through. What do you have to do in order to get ready for that sharing to happen? And that can help you have a plan, and having a plan takes some of those emotions, that stress that we talked about, out of your data management. Metadata is data about data. So this is for all of the data sets you might build in your research. You want to make sure that you are also collecting information about what the units of the data are, context of the data, how it's created, sources of the data, and a shortcut to thinking through some of that metadata is thinking through like a repository, a data repository it might end up in. If there is one related to your discipline, that can give you a nice shortcut to the types of metadata you should be collecting based on what similar data sets have collected in order to make their data more understandable. So just a reminder, feel free to ask questions in the chat or in the shared notes document about anything that's going on, and also use those spaces to share your favorite tools and tricks that you found to help you on any of these topics as we go through. So just briefly, one of the data management things to think through at the beginning of a project is the development of a directory structure that can keep your project organized. This is just one example of what a project directory structure could look like. It really helps to keep your research materials together as much as possible in subdirectories like this. Because if you create the structure from the beginning, then as you're doing the work, it's just a matter of saving things in the appropriate place, which means finding them later is much, much easier. The other thing to keep in up to date in your directory is a project level README file. You might end up creating multiple README files as you create your project, but you should have one that is at this project level that will describe like essential information about the files in this directory. So this can be a very adaptable piece of documentation. You can put in context about the work. You can outline the different materials and how they relate to one another. Data types, data formats, any citations, including data citations. This sort of thing can all go in your README file. And then once you've created the structure, it becomes a lot easier to stay organized, because when you're saving something new, you just put it in the right place, and that is actually providing context to someone else to understand what that file is. And so one thing to keep in mind is to always keep your raw data, and to always keep a read-only version of your raw data somewhere safe and backed up. So if you're going to be cleaning up your data, processing it in different ways, that's great, but keep a read-only version of the original raw data set untouched, so that you can always go back if you need to and see what was actually done, which is often a barrier to reproducibility, is losing that life cycle of the data over time. Always back up your data. If you've been in research for a while, you've heard horror stories of people losing what they worked on for a year, 2 years, 3 years, through a server breaking down, a laptop being stolen, a notebook being misplaced. So the rule of 3 applies here. Aspire to have your materials backed up in 3 places, 3 unique locations. An example of that is to have it somewhere in the cloud, somewhere on a server, and then in another third place, it could be a personal drive, or maybe you have a hard copy somewhere. So that's the data management at the project level. And then one of the things that I found very helpful is when you are starting a new project, to think in advance how you're going to name the files that you create in a way that makes it easier for you to identify what that file is down the line. So a file naming convention, a very simple one, is a great way to get started with that. So when you come up with a file naming convention, the rules themselves don't matter as much as the fact that you have rules. So keep it simple to start. Here are some of the things to keep in mind. If you're going to put a date into the name of your file, you want to do it year, month, day, because that allows a computer to sort the files as they're created in the appropriate order. Use meaningful abbreviations. So think through what are the different variables in what you're collecting in your research, and you can put those into the name itself to allow you to sort through your files that way. Similarly, you can have group identifiers if you have different groups, and I'll show you what I mean in a second. And as you come up with these rules, pick a couple to start with, document them in your README file. be consistent and think about how you're going to track versions over time. So you can use version control, which is something that can be built into a platform that you're using, such as an electronic lab notebook, or you can use numbers such as 01, 02 to say this is earlier and this is later. So this is what I mean when I talked about abbreviations and group names. So this is just one example, and mind you, it's a very long one, so most of you are not going to have file names quite this long. But what you want to do as a general rule of thumb is go from general to specific from left to right. So the date over here, for example, you want to delimitate these different sections of your file name with something such as an underscore. This is a project name. This is an experiment name. This is the data type. In this case, this is the ID of the individual that collected those data. And here we have a version number. So this is just an example of what we're talking about. But this in and of itself can go a long way in taking away some of that stress that we talked about when it comes to managing your data. So I'll pass this back to Nafisa. And again, remember, you can ask questions and I'm happy to answer them during or after the workshop.
Great, thanks so much, April. And April posted in the chat, the website that I was talking about is Retraction Watch. So they're an interesting website to monitor in terms of all the different retractions that are occurring. Okay, so let's talk about electronic lab notebooks. How many of you, if you want to just raise your hands or whatever, use an electronic lab notebook in your research? I see some. Okay, cool. Awesome. So we, again, like April mentioned, we have a little bit of, we have this online note document, so feel free to add tips and tricks of what you like about your lab notebook if you do use an electronic one. So next slide. So we know that lab notebooks are really important. They've been used since the 15th century. They're important for dissemination of ideas, research findings, documenting what you did. So when you go and you write up the paper or your thesis, a year, 2 years, 3 years, 4 years down the line, you have some record of what you've done. In terms of industry, sometimes these are, and academia, they can be legal binding records that protect intellectual property. And we can see an example of a lab notebook here. Traditionally, they've been thought to, be actual physical notebooks. So hard copies, if you can see a copy of, or a page of Leonardo da Vinci's lab notebook. And the problem with these hard copy lab notebooks is that they're not searchable. We can't, type in Western blot or the date of a Western blot looking at an autophagy marker. That's not possible. These hard copy lab notebooks can be damaged. If there's a fire in the lab, if you spill, a solvent on them, or they can be lost. So there's no way to really, and there's no way to back them up. And if you have collaborators, it's hard to share components of the lab notebook. It can make it all very tricky. So there has been this emergence of lab notebooks or electric lab notebooks. Next slide, please. And these notebooks are really the ELNs, or the electronic lab notebooks, provide a way for you to search both online and offline, as well as exporting data, sharing data. You can embed figures, so pictures, protocols, and then there is this global accessibility in terms of if you have a connection to the internet, you can access them from anywhere. And some lab notebooks also have mobile apps, so you can, update things on your phone. And so it makes it really accessible. And so these are just some of the highlights of what an electronic lab notebook can do. And we're just going to walk you through, next slide, please, of what some of the basic features are of an electronic lab notebook. So you have, a way to search things. You are able to, like in this image here, we have a gel, and you're able to embed that figure. You can share things. You can have, different subfolders that we see on the left here in terms of different projects, different components of a project. You can organize things as you need to. There's also data entries are dated. You can, export things to print, to share, and it makes it a lot, it makes research a lot more easier in terms of being able to share. What I found really great with electronic lab notebooks is that we can have all the protocols for the lab in one place, and everyone has access to them. So I feel like it makes things more inclusive in the sense that not certain people are, having certain protocols. All the protocols are in one place, they're updated in one place, so everyone can see everything. And I really like that in terms of my lab, and people are able to see what other people are doing or how they did it in the past. And so there is this sort of knowledge that's being passed on. And so there are a lot of products, a lot of electronic lab notebooks that have emerged in the past few years. So there are some things, to keep in mind in terms of consideration for the different products. So there are options where you pay for the lab notebook. And then there's also free versions, there's open source. Some of these paid versions of electronic lab notebooks for academic research, they sometimes waive that fee. I currently use LabStep, which is based out of London in the UK, and they do not charge for academic research purposes if you're using their lab notebook for that. But there is this great resource that's been compiled at Harvard University. Next slide. It is a Google Sheet where you can see all the different lab notebooks, electronic lab notebooks, and the different components each lab notebook has. And it's really well organized. So if this is something, that you want to get involved with or incorporate in your research, this is a great resource in terms of the different features that are involved for each lab notebook. Okay. I'm just going to check the notes. Okay, great. So in terms of just general tips for if you're incorporating or thinking about incorporating an electronic lab notebook is, backing up the data regularly. Perhaps maintaining a physical notebook as well in addition to your electronic lab notebook. And this again depends on your type of research that you're doing. If you are moving around, if you collaborate and travel a lot, you might want a lab notebook that has an app that's on a phone or on a tablet. When you are considering a free electronic lab notebook, you want to also check the privacy and data ownership as well. That's an important consideration. Great. Okay, so now what we're going to do is we're going to go on to protocol sharing and organizing. So so we know that when I remember when I started in a lab and trying to, figure out protocols and optimizing them or finding a protocol that was working can be quite tricky. And so, this is, next slide, please. Finding a protocol that is working or somewhat working in your organism, in your tissue of choice or your model organism can save you a month, sometimes even years, sometimes depending on what your area of research is. And it's important to, we want to accelerate science in terms of this workshop. So there are tools out there where you don't have to reinvent the wheel. And so there are many places to find protocols. Next slide, please. You can get them from lab members. You can create them yourself. oh, can you just, oh yeah, you can get them from the literature repositories. There's the website ResearchGate or contacting people in the field. So in terms of literature, next slide, we can see, I don't know how many of you have ever experienced this where you've read a paper and you've like, oh, I'm going to use the protocol in this paper. So you go to the methods section and then you find that it cites another paper. So we have this tweet here, and so you're looking for a protocol in a paper that was published in 1997. And it says, as described in X et al. 1996 or reference number 96. So you go to the 96th paper and you see that it's described in this paper from 1987. And then you go to that paper that was published in 1987 and you hit a paywall. So you're not really finding this protocol, it's kind of just like, wow, you're stonewalled. And so it can be quite frustrating. Next slide, as shown by this, just tucking the computer out the window because you really need that protocol. And so there's also, when you go into the literature looking for protocols, the description can be ambiguous. And you can see that here in this tweet, in terms of looking at some sort of device that was trying to replicate the making of this device. And you go back to the original reference in 2009, and the devices were fabricated with conventional methods. What does that mean? How are you going to replicate that? What are the details there? And so this can, next slide, can be quite frustrating for an individual. And so, next slide, ambiguous protocols are often difficult or impossible to reproduce. And so this is this how to draw an owl joke you see this beautiful owl that's drawn here in step two, and then how to get to that beautiful owl or that beautiful scientific result. And so the instructions are draw some circles, but that's not all that's involved in making this owl drawing. So this can be quite frustrating, but there are resources that have been developed to help with protocol sharing. So some examples are, next slide, are repositories. So there is this push to replace journal supplements, so method sections that are often posted online, with repositories. So there are several repositories, next slide, that have been developed. There's a protocol exchange with Nature Research, protocols.io, which is one that I often use a lot. PLOS One also has a protocols repository. And so these are websites where people will share, their protocols that they have developed in different cell types, different organisms that you can look up and, get some sort of starting point. And these protocols often have, these repositories have different requirements, catalog numbers of products, steps, and all of that in order for the work to be able to be reproduce. And so you don't need to reinvent the wheel. And so next slide. So they're just an example of the power of protocol repositories. So there was this tweet posted by this scientist that's looking for RNA extraction from primary cortical neurons. And then next slide, and then you get a response to this tweet where there is a link to a protocol on protocols.io. And this protocol has been published in a repository, so protocols.io. And the scientist replied, and it worked out really great for him. And so next slide. So these repositories build this environment for these protocols and the easy sharing of these protocols. And so we see next slide that they are a way to accelerate science in terms of making things reproducible, facilitating scientific or research connections, the reuse of things that have been developed, and enhancing the value of research. So not spending 3 to 6 months developing and optimizing a protocol, but starting at least from somewhere. Okay, so we're going to do a quick little exercise. And so if you guys want to pull out a piece of paper or a pen or some sort of writing utensil and writing surface, whatever you have available, or if you're on your computer, just I don't know, whatever. I'll give you a minute and then we'll do this protocol a drawing exercise. So what I'm going to do, or what we're going to do, is we're going to give you a bunch of instructions. So we're going to give you a protocol of to draw something, and then we'll see what we get. Okay. I'll just give you a couple more seconds to find something, and then yeah, any questions so far? Or all right, and feel free to add your questions to our notes document. So next slide. So we'll share this protocol with you guys. So can you just go through steps 1 to 9, each step, and do each step, and then you'll come out with something. And yeah, so we'll give you a few minutes to do that. So April posted in the chat box if there's any guesses on what this is supposed to look like. So feel free to pop in your guess.
And face is a very good guess. It means what you drew is probably closer to what I drew the first time I did it. Scribbles is accurate. Does it look like anything that anyone's seen before? It sounds like maybe not. Mickey Mouse. A cell?
All right. Should we give him one more minute, April, and then we'll be about
Yeah. Give him one more minute.
Yes.
There's more than one Mickey Mouse. Oh, Jose.
All right, we're at well, a couple more seconds, and okay, so we'll just go to the next slide, and you guys can see. So Jose hit the nail on the head. It is supposed to be SpongeBob SquarePants, and you can see we have some examples of the different drawings. So we had one protocol, but it resulted in several different types of results. And then the next slide, you can see SpongeBob SquarePants here, and then all the different ways that he was drawn using this protocol. So hopefully this kind of, hits the nail on the head that one protocol can result in a number of different results. And so in terms of, getting access to protocols when you're starting from scratch or starting in a new area or looking to optimize protocols, there are these repositories. And in terms of if you're wanting to, digitize your protocols, these are some tips that we have. Next slide. For thinking of how to digitize your protocol if you're leaving your lab, or if you're moving on to a new project, or if you're just archiving material in your lab, thinking of the protocol as being this self-contained publication. So, including a couple of sentences about the context of the protocol, the methodology, and then including as much detail as possible, the time for each step, the reagent amount, vendor name, catalog number. I remember times when I was in graduate school and I was like, what is the antibody catalog number? I want the same one. Why don't they list this in papers? And there's also expected results, any safety information, the steps that are involved, any other tips or tricks. Sometimes the moon has to be facing in a certain direction for scientific experiments to work. So there are great resources here. Protocols.io is a great resource, Google Docs, electronic lab notebooks. So you have this sort of versioning, tracking of your protocol as it's getting updated and changed. And I also just wanted to mention there are protocol journals that are available to, like Jovi, bioprotocols, that also include some of this information as well in terms of getting protocols up and going. Okay, so I'll pass it back to April now.
Thanks, Nafisa. So just to continue this sort of going through how to share and document the work that we're doing. Another side of this is the sort of materials that you're using in your research. For example, reagents and other types of research materials. How do you document and share those in a reproducible way? So some of the problems that we see related to reproducibility and reagent availability is oftentimes people will publish a paper that says that their materials are available upon request. But as we all know, people move from institution to institution, they move on to new projects, and where that reagent or other material that's needed actually is located, sometimes that information gets lost over time. So even if there is a request coming in, even though there's an intention to share it, it's not always possible as time goes by and records are lost. So how we know this is because there's been multiple studies on it. This is one that the NIH found a while ago. They found that the half of the mouse lines had to be remade at least twice. And in this study of trying to access these research materials, only 12% of the papers that they reviewed in this study had their mouse lines available in repositories. And you can see here that it's very common for a lot of labor to go into the remaking of these materials over and over again. And this is a waste of people's time, and it's a waste of their money, and it can result in things not being redone the exact same way as they were the original time. So it's important to think about these things in terms of like, oh, well, if we think about how to do this from the beginning, how to share these things from the beginning, it can feel like extra work at the beginning, but what we need to keep in mind is it can save us and other researchers a lot of work down the line. Another example of a problem with access to the materials of other researchers, there was a plan to replicate, it was a reproducibility project to replicate 50 high impact cancer papers. And they had to shrink that very, not a huge number down to 18 because they weren't actually able to access all the information and the materials from the original authors. And these are big papers that have been cited many, many, many times. So this is a problem, and it's something that reagent sharing can really help with. So for the sake of time, I'm going to skip through this. It's a just general outline of what are the barriers to the current system of just asking for reagents. There's a lot of labor that goes into the work of sharing a reagent for a study that you did. So using a repository allows you to put that into the hands of a professional, someone who will take responsibility for managing requests for those materials over time, ensuring that they're preserved properly, and it takes it into less of the work of an individual author and more into the work of the scientific community at large. So it also means that these materials might be available more quickly when they're requested. It means that the quality of the materials that are shared are authenticated, so you know you're actually getting what you think you're getting. It allows people to curate them so that it's easier for people to discover the materials that they need because they've been organized in a way that makes sense to other researchers. And the like nitty gritty of shipping these things, making sure they're shipped properly and they're tracked and they're insured, all these things can be aided by using a repository. So when you're thinking through your research, you want to think about documenting the things that you need to document in order to allow these materials to be shared in your papers. So you want to track how all your materials are created, how your reagents are created, authenticate them, make sure that you think you're using what you think you're using, provide citations to the papers you referred to when creating them, use descriptive and standardized naming conventions if you're naming a reagent, record and include any of the catalog numbers or research material ID numbers, in all of the associated writing that you create, such as a protocol, your notebook, your manuscripts. Make sure that these are written down somewhere. And then finally, put them in a repository. So what repositories are available? Okay, so I'm going to go, because we're running a little low on time, this is a little bit up more on the benefits of depositing a reagent, that just highlights that there's some evidence that folks will cite your paper more if they're able to access your materials and therefore replicate the research for themselves. So there are some benefits to using a reagent repository. And here are some examples of what we mean when we say reagent repository. Adgene is a nonprofit reagent repository commonly used. It covers a lot of different types of materials. And then depending on what types of organisms or tissues that you are working with, you can see here that there are often specific repositories and biobanks for the materials, those specific materials. They have the expertise to ensure that those are being stored and authenticated properly. So in summary, sharing your reagents can increase access, and it can speed up how quickly someone else can reproduce your work and therefore cite you. You want to make sure that you're recording details as you're doing your research, so that that metadata can go into the repository when you share and into your paper when you publish. Deposit the reagents that you make. This can improve reproducibility and help collaborate with other researchers who want to build upon your work. So data and code sharing is going to follow a very similar pattern to what we've talked about before. There's a theme here, which is that planning to share your work in a repository can help you think about what documentation you need to create as you're working. And also, repositories are a great way to share your research materials in a way that gives you credit. So when you're thinking about sharing your data and code, which many of you are going to, if you haven't done it yet, are going to be required to do in the future, it shouldn't be something that should be too overwhelming. There's just a few questions that you need to think through. First of all, you want to think about what you want to share. It's important to share any of the materials that are necessary to validate the findings that you're publishing, as well as to reproduce the results so they can be built upon. You want to share any data and code that could be valuable to other researchers and also policymakers. So there's a lot of different stakeholders beyond just researchers that might want to reuse your data and code. And then also any data and code which is not easily regenerated. So sometimes there's a data set that was built either over a long period of time or in really rare circumstances or expensive circumstances. And we don't want to be, as a community, having to remake the wheel over and over again. So these are good indications that you should think about sharing those materials. In addition to that, there's an increasing number of mandates, sometimes from funders, sometimes from publishers, for sharing these materials. There is some evidence of a possible citation benefit for papers that have their data accompanying their published paper. But I would say the main motivation that you should keep in mind when thinking about putting your data in a repository and putting your code in a repository is the best way to not lose it. Oftentimes you want to come back to something a few years down the line, and so much has changed in terms of how you work that actually locating all your materials and your data can be difficult. So when you finish a project and deposit it in a repository, that preserves long-term access to those materials you created in your in your research, such as your data and your code, it preserves the access not only to other people that are going to read your paper and try and build on your work, but it actually preserves access for you and people in your lab to have access to that over time. And in general, when you're thinking about how to share those materials, choose open, persistent, and non-proprietary file formats so that they can be reused easily by people without having to purchase a license, and are more likely to last over time, that people can open them in 5 and 10 years. Make sure you share the documentation. We talked a little bit about that before. Include citations to any source data or code, and make sure you have metadata about your data and code that allows people to understand what is in the repository. So it's important to share these materials in a place that doesn't disappear. So this is a chart showing some links in different published abstracts of scientific studies and how over time those links stop working. So we want to put our research in a place that is persistent and preserves access. So don't put it on your website. So we want to choose repositories. Repositories allow you to create a persistent identifier that is associated with your data and your code. A DOI is an example of one of those. It's unique, it's citable, and it won't break. It will always resolve back to an instance that should contain your data. Repositories have preservation plans. They back up the data sets that they store. They have a management of access, so you can allow certain people to see certain things. They allow versioning, so if things evolve over time, you can always keep them up to date, and it allows you to pick a license, and that allows you to tell the people that are coming to your data sets how they're allowed to be reused specifically. So I won't go into licensing right now, but this is just to give you an example of how to start thinking about which licenses you might pick for your data and your code. There's some good sources there to think through the different types, and your local library at your institution is another good place to talk to about what licenses are appropriate for the materials you're making. So the commonly asked question we get is, where should I put my data? Where should I put my code? So we've broken this down into 3 different types of repositories you want to think through. The first one you want to think through is whether there's a place you have to put it. So sometimes a funder will mandate that your data need to go in a certain repository, or perhaps your university mandates that your materials need to go in a certain repository. So those are a head start. That tells you where you want to at least make sure your stuff is in there. If you don't have one that's mandated, the next place to think about putting your data is in a domain or discipline specific repository. So if you're in a particular type of research, then the people that are in that type of research as well, that discipline, are going to be looking in that repository for the types of materials that you create. And it also will be likely a repository that is built to actually be able to preserve and properly store that those materials. Like the size is probably the same, the format is something they're familiar with, as well as the metadata. They'll be familiar with the types of metadata that your colleagues are going to look for when they are access your materials. So if none of those actually meet your needs, there are general purpose repositories. Some of them are free. Some of them are free up to a certain file limit. And these are places to start. Zenodo, I would say I would give an extra little check mark myself. I've had really good success using Zenodo. It accepts a wide variety of types of data sets. It's free, it's pretty generous with the data set limit, the metadata is great, and it integrates with a bunch of tools. Oops, I didn't mean to open Zenodo, but there you go. One of the benefits to using Zenodo is that it's integrated with GitHub. So we mentioned how you should share code that is necessary to reproduce your research. So you can store that, as people often do, in a platform called GitHub and integrate that repository with Zenodo. So you have a release in Zenodo that gives you a DOI, so you can be cited for the code that you're sharing. So some principles to follow sort of at a high level when you're thinking about sharing your data is called the FAIR data principles. So we won't get into the very large amount of literature written about FAIR. The thing that you want to know as someone who's starting to think about this is just that FAIR stands for making your work findable, accessible, interoperable, and reusable, and to strive to do that whenever you share a data set. Nafiza, on to you.
Okay, so we're going to cover I think some really important stuff in terms of dissemination of data. And so this is next slide, data visualization and analysis. I actually give this workshop at a lab meeting every summer in my research group, and I really like this part of the presentation as well as all the other parts. But in terms of stressing the importance of sharing scientific findings. So next slide. We know that data presentation is the foundation of scientific knowledge, and that comes in the form of figures, because they often show data of key findings that we have. And so next slide, we know that it's really important to have effective figures, and these figures should convey information about the study design. So, time points, control groups, they should illustrate important findings. And I think the biggest point here is that you want to create figures or tables, data presentations that allow your reader to really critically evaluate your data. Because I think, in terms of when I'm putting data together, I want feedback on my data, and I want to engage the person that I'm sharing the data with so that they, are able to, ask questions that are applicable. And sometimes questions can be terrifying, but I think it's a really good opportunity to get feedback on data. And so creating effective visualizations is really, really important. And there's a reference here to a paper by Tracy Weisgerber that I always point my students to read. And I would also point that out to you guys as well. It's a great paper, and we're going to go through different parts of the paper in terms of thinking about your data and how to visualize it. Okay. So next slide, there are a couple of ways that people like to use or share their data or organize it. So these things, so we've highlighted them here: Excel, Google Sheets, Open Office. And so, there are some issues with these programs in terms of macros and dashboards and reducing reproducible workflows. The default plots for these programs are often bar charts or line plots. And so that can be a little bit tricky in terms of thinking about how to present your data. So next slide. Like I mentioned earlier, you really want to show your data. So here we have a cute little graphic where we have on the left, we have a bar chart, a bar graph we have some sort of variance. And, this is showing possibly mean and some SEM, standard error of mean or standard deviation. And then on the right, we see all the data points as well as this summary statistic. And what we can see is we can see the spread of the data. We can see how many samples were in this data. And so you can see that you're, already engaging the person that's reading this graph or this little cute cartoon in terms of understanding what's happening. And so next slide, it's, you want to be a little bit critical when you're sharing summary statistics. There isn't really an alternative to showing the actual data. Next slide. And we can see here, just some alterations with the summary statistics. And so what, next slide. You can see here where you have on the left the reader as a passive observer. So you have the mean and the SEM of whatever data is being presented here. And then on the right, you can see that the reader, is able to become an active participant. You see where the samples are for each group in terms of the spread. In the third group, from the y-axis, there may be an outlier in that group. You can see that not all the groups have the same number of samples. That might be something important to take in terms of interpreting this result. And so hopefully this shows you the importance of actively engaging whomever you're showing your data to. Next slide. So this is a figure or a component from the paper that I recommended in terms of looking at recommendations for how to present data. So bar charts are often not thought to be ideal for continuous data. And you can see that here in terms of how the data is represented when you have outliers, when you have bimodal distribution, when you have an unequal n on your right side here. And and yeah, the statistics might be significant or non-significant, but how are you engaging the reader to interpret this data? Okay. And then in the paper that I recommended, that's also cited down here, the next slide, there's also a really great section in how to choose the right plot. So, there's dot plots, box plots. I learned about violin plots reading this paper, when to use bar graphs. And so what Tracy has done and her co-authors have laid out recommendations for, how to actively engage your observer when you're sharing your data and what type of plots can be most effective. Okay. And so let's talk a little bit more about these dot plots. These dot plots are where you're showing individual samples. And so making effective dot plots is shown here in this slide in terms of, showing all the groups, looking at summary statistics and things like that, and what tips can be done. Next slide. So making all the points visible, emphasizing summary statistics. So having the bar, the mean or the median, whatever you report in your findings, maybe some sort of variance, SEM or standard deviation, making it easy for the reader to interpret your results. I always view when I'm putting together talks or papers, it's my job to make it easy to understand and engage the reader so that I can get effective feedback from reviewers, from people that are attending my talks. And so this is one way to do that in terms of showing your data. There's also this really cool website where, next slide, you can actually have interactive plots. So this might be something good for if you're giving a talk. So there's interactive dot plots or line graphs. So getting, sort of that animation involved in your talks. In terms of products or things that can be used, tools that can be used to create these effective graphs. We have some recommendations. Prism is a great program. It is pricey if you don't have institutional access to it, but it is a great program for making graphs. There's also other options. Tableau. R is a great, if you're into programming or know how to program, that's a great program as well for creating effective graphs. I'm just going to check this. Okay, there's no questions on the notes. Okay, so just to wrap up our workshop, we've come to the end. We hope that we have shared some of these tools in order for, next slide, for you to increase your reproducible research. So in terms of organizing experiments, analyzing results, sharing your results with researchers, sharing techniques, reagents, and in hopes to help you accelerate science and contribute to the big picture and all of that. So take what we've learned and if you're able to apply one thing to your research, that's amazing. And that's that initial growth of becoming a scientist in terms of increasing your reproducibility. And possibly some of you, I think, are already implementing some of these tools in your research, which is great. So in terms of next steps, it might be something that you guys want to ask yourself. What is one thing that you can do today to make your research more reproducible? Feel free to add to our notes document if there's something that you want to share with us in terms of making your research more reproducible. And feel free to check out our website. We're always looking for people to lead workshops or get involved in our community. So if you're interested, please check us out. We're also, like April mentioned, doing a crowdsourcing fundraiser right now. So she'll send the details of that. We have a lot of funders, next slide, that we'd like to thank. Our current funders, Chan Zuckerberg Initiative, AdGene, as well as our past funders, Protocol.io, eLife, the American Society for Plant Biology, Mozilla. And next slide, like I mentioned, all of these materials are published on our website under the Creative Commons with Attribution license. And then next slide, we're here, April and I are here to answer any of your questions. We'd also like to solicit your feedback. So please take a moment to fill out this form. Yeah, it's been really great spending this time with you, and hopefully, yeah, we can answer any questions you guys have. Feel free to put your question in the chat box or to unmute yourself if that's possible.
So you can also feel free to reach out to us anytime after if questions come up, after the fact. As Nafisa mentioned, this is a community-led group. So if you are a scientist that is interested in reproducibility, or maybe you have an idea of something we can add or change in the workshop, all of the content is created by the community. So reach out and let us know.
I have a question, if that's possible.
Yes.
How you mentioned before that the students used electronic notebook. How is the student buy-in percentage in using such notebooks?
So in my lab, we just use electronic lab notebooks. And sometimes students, it takes a while to learn, but most of them are more tech savvy than I am. And they just love it. It's really easy to update. And they're able to access everything. I haven't had any one sort of complain or say, I want a physical lab notebook.
Mm-hmm. And do you review the protocol with them together after?
Yeah, so we'll discuss results and we'll discuss protocol changes and any questions that they have. I try and meet with all of my students on a regular basis to make sure that they're, getting everything that they need, or if they have any questions or anything, for sure.
Mhm. Thank you so much, Nafisa and April. That was really helpful. I'll definitely join in the community. I'm very interested in reproducibility. So yeah, thank you so much. That was really helpful.
Awesome.
Awesome. Thank you so much for coming.
Any other questions, comments? Don't be shy.
Also, feel free to share things that you found that helped.
It looks like there's a question in the chat.
Do you have a schedule to update the ELN, Nafisa? Do you make a schedule?
No, we kind of update I do check my progress of my students every week. I have kind of a reminder that comes up at the end of the week. And then as protocols are changing, we just update on a case to case change. So if we're working on a study and the protocol has changed, we'll make sure to update that and get a new version of it updated. So it's kind of just like a living document or electronic component. Does that answer your question? Please let me know if it doesn't.
Yeah, it does. I actually had a little more follow-up. Like, I often feel like when I am trying to create notes, I don't know what to write. Like for example, like I don't work on wet lab stuff, so it's not like I have, okay, I'm using this reagent, but like when we are actually doing experiments, we make very like detailed notes, but that doesn't go into the electronic notebook. So like when it comes to coding stuff, I have my own like, whatever I do daily kind of thing, but I don't like a diary of sorts, but I don't know what to put in an electronic lab notebook, honestly speaking. And I'm a grad student, so I want to be more organized, and I'm just trying to learn. If you have any tips?
So, I've always used paper or a hard copy notebook for all my training. And then once I started my group, I was like, okay, let's go to electronic lab notebook. And I remember being frozen. Like, what do I write in here? And, I just treated it like a paper notebook. So I write the date, or it's already dated, and I'll write what I did, or, the experimental groups and dilutions of antibodies, or whatever it is that I'm focusing on. So whatever I would write in my paper notebook, I write now in my electronic notebook. And I do my lab staff, what they will do is every day they'll write what they did and document like a diary.
Got it. So, okay, so basically, I kind of have a habit of creating like a PowerPoint presentation for my daily stuff. Like, that's just something I've been doing, but I guess I could instead like use the ELN. But yeah, I think like I'm halfway through I'm more than halfway through my grad school, so I'm really scared of changing things around, but it's always about updating. Also, I would be interested in being part of this as well. This is so great. Thank you so much for your time, and all these resources are wonderful. Thank you.
Yeah, check us out. Feel free to go over to our website, and yeah, April's done a really great job of keeping it maintained and all of that. And yeah, we'd love to get more people involved.
Absolutely. And it's so common what you said about you're halfway through something, so you're worried about changing it. And I think this is really common. And I think don't, like, if something is working for you, don't feel pressure to go and change how you're doing everything in the middle of a project. I feel like when a project comes to an end, that is the time to think, now, what is the one pain point I had in that last project for organization or documentation? Now, what is a way to make that less painful for me in the next project, and make one change? So that's always what I tell people to do. It's like every new project, you try and do a little bit better. And over time, we just end up being more organized and more reproducible.
Thank you. That makes so much sense. I really appreciate it. Thank you.
Well, I just want to thank Nafisa and April again for coming and sharing their time with us. And also thank you, Joanne from GPS STEM, and collaborating with ICTS to put on this workshop. So thank you, everybody. We'll circulate the slides and recording once it's ready. I probably should have mentioned this earlier before too many people jumped off, but the ICTS also has a survey for you to fill out. So we'll be sending that out a little bit later. Thank
Awesome.
you so much.
Yeah, thank you so much, everyone at Marguerite and the whole team, and Harinder that helped make this happen. And I look forward to meeting those of you that come over to R4E to run a workshop.
Thank you.
Bye.